So we're going to pick up here with Thomas Hunt Morgan and think about Drosophila, his fruit flies, and the idea of linked genes. So Thomas Hunt Morgan, like we said in class, was a critic of chromosomes being the basis of inheritance, and then he ended up winning the Nobel Prize for, in fact, figuring out that chromosomes were the basis of inheritance. And he did all of his work with fruit flies. Scientific name is Drosophila melanogaster. So quick review of some of that vocab again. Gene is just a sequence of DNA, a chunk of that DNA on a chromosome, A's, T's, C's, and genes that code for a strand of messenger RNA that's going to go code for a protein. Then that protein is going to have some kind of effect on a phenotype, on a trait. Locus is just the location on the chromosome where we find that gene. So what chromosome and what location. Homologous chromosomes, these are the chromosomes that we get from mom and dad that have the same genes, but not necessarily the same alleles. I'm actually going to skip down to sex chromosomes and then go back to autosomes. So sex chromosomes, these are our X's and our Y's. So humans with normal chromosomal composition are going to have two X's or an X and a Y. Which will determine sex. And then those autosomes are the other 22 pairs of chromosomes that aren't X and Y. It's just our normal, everyday, boring chromosomes that code for everything else that we are. Super important, but maybe not as exciting as those X's and Y's. And then independent assortment is just that idea that our parental chromosomes, those chromosomes that we get from mom and dad, do not follow each other during nuclear divisions. And so when we go through meiosis and we're making eggs and sperm, we don't make eggs that have all of mom's DNA and eggs that have all of dad's DNA, but instead, statistically, we're more likely to get a pretty good hodgepodge combinations of the two. So Morgan, again, was working with Drosophila, and he noticed a white-eyed fruit fly in his population. And so he decided to cross that weird white-eyed mutant fly with some normal flies and notice that most of the offspring were red. And when we do this, we're usually going to do big R for red and little r for that recessive white. But Morgan did a little bit different. So he would use some letter with a plus for the wild type, for the normal trait, and then no hashtag, no whatever would be that mutant. IB wants us to know this version. This is what you'll see on quizzes and tests. This you might see in college. And so I like to throw it out there just so that it's noodling in the back of your brain. So here's just showing again that we've got these test crosses where I've got some wild type and then mutant. So this organism is a heterozygote for this trait, gray body, and then this one is for his normal wings instead of vestigial wings. And so you end up with these organisms, and again, like we talked about with our test crosses, we always use homozygous recessive, or in this instance, we're talking about a mutant. Good. So just seeing again those ideas, that notation that Morgan used. So again, Morgan mated his heterozygote red-eyed flies, and we would show like that on our Punnett square. Whoa, sometimes this gets a little out of control. So our Punnett square would look something like that, and a fourth, our white-eyed, makes total sense because there's our 1/4. 1 out of 4 genotypic ratio of homozygous recessive, 1 out of 4 probability that you're going to have white eyes. Makes total sense. But the weird thing was that all the white-eyed flies were male. And so Morgan went back and started to think about what could have caused that, what could have linked that white-eyed trait to the sex of the fly. And he decided that gene for eye color must be on the sex chromosomes. And so we have this idea of things being sex-linked, genes that are simply on those sex chromosomes on the X's and Y's. So this idea of linked genes, again, genes are linked if they're on the same chromosome. And we can have sex linkage where we've got, this is chromosome number 1, this is the sex chromosome of fruit flies. And so these are some of the genes that would be linked to the sex of the bug. And then here are the autosomes, these other 3 chromosomes. These are the autosomes, and they can still be linked. And so being eyeless and or having sparking eyes is linked on chromosome number 4. So these are simply traits that are more often inherited together simply because they're near each other on the same chromosome. So this is just showing another picture. So instead of calling this big R and whatever, well, it would be X and Y if we're looking at those sex chromosomes. This is just showing you a copy of the X chromosome and a copy of the Y chromosome. And you'll notice the Y chromosome is missing some genes. So there are a lot of genes on the X chromosome that are not on the Y chromosome. And we talked today in class about this idea of being hemizygous, H-E-M-I, hemizygous. So instead of being homozygous or heterozygous, you're only half because they only have 1 copy of the allele instead of 2. They only get 1 from mom because that Y chromosome doesn't carry a lot of genes. So in humans, hemophilia and color blindness are both considered, well, not considered sex-linked. They are sex-linked. And so we would use that same kind of notation, that sex-linked notation to show that. So if a colorblind man, so he's going to be XY, and we're going to show that he's colorblind with a lowercase c, since that's recessive, has children with a homozygous dominant woman, so she's going to be XX, and she's got totally normal vision. So we're going to give her big C, draw our amazing Punnett square, and we've got big C, little c on our X's, big C and a Y on that young man, big C and a little c on those X's, big C and a Y here. So our genotypes, we're going to have 50% XX, big C, little c, and 50% X, big CY. And then in terms of phenotypes, we're going to have 50% girls with normal vision because they've got that dominant allele, but they're carriers because they also have a copy of the recessive. So I've got 50% females with normal vision, but they're carriers, and then 50% boys with normal vision would be the genotypes and phenotypes. So hemophilia is also a sex-linked trait in humans. So normal man is going to be carrying the normal proteins for blood clotting, and the woman is a carrier. So she's going to be heterozygous. So we draw our Punnett square. We've got XX with big H, XY with big H, and nothing on the Y. We have XX with big H, little h, and we have XY with a little h. So genotypically, we have a bigger spread. So there's 1 out of 4 totally normal, not a carrier female, 1 out of 4 normal male, 1 out of 4 female that's normally phenotypically, that has a normal phenotype. She can clot her blood, but she is a carrier. And then we have that 1 out of 4 male with hemophilia, so with that blood clotting disorder. So genotypes, 4 different ones, and then our phenotypes would also be 4 different ones. So normal female, normal male, well, actually, I guess we would probably call those the same. Since she's just a carrier, she's still going to have her normal blood clotting. So 50% normal female, 25% normal male, 25% hemophiliac male.